Hey everyone, and welcome to The Explainer. Today, we are embarking on a truly fascinating journey. We're going to look at the literal recipe of life itself. We're starting way down at the most basic elemental atoms, and we're going to work our way all the way up to the magnificent, highly coordinated molecular choreography of protein synthesis. Seriously, if you've ever wondered how a bunch of lifeless chemicals suddenly organize themselves into the complex, breathing, thinking machinery of a living cell, well, you are absolutely in the right place. Let's get into it.
So here's our roadmap for today. We'll be covering the essential solvent, energy and storage molecules, the protein building blocks, the genetic instruction manual, and finally, the protein synthesis factory.
Section 1. The essential solvent.
You know, it's really easy to just think of water as, well, just a liquid that fills up space. But biologically speaking, it is the foundational stage for life. Without it, literally none of the reactions we're about to discuss could happen in your body. Now, to really understand water's magic, we first have to understand its polarity. Polarity simply means a molecule has a partial negative charge on one side and a partial positive charge on the other. Because of this uneven distribution, water acts kind of like a tiny magnet. And as we'll see, this uneven charge changes absolutely everything about how water behaves. Check out how this polarity plays out in real time. The slightly negative oxygen atom of one water molecule naturally attracts the slightly positive hydrogen atoms of the water molecules next door. This attraction creates what we call hydrogen bonds. Now, by itself, a single hydrogen bond is pretty weak, but when you have millions of them together, it creates this tremendously strong dynamic network. Because of those hydrogen bonds we just looked at, water gets some incredible, almost magic life-sustaining properties. It acts as a vital solvent, pulling apart and dissolving ionic substances so chemical reactions can actively happen inside our cells. Its high specific heat capacity means it takes a ton of energy to heat it up, which naturally buffers our internal body temperatures. When you step outside and sweat, its high latent heat of evaporation cools you down incredibly efficiently. Then there's cohesion, water molecules sticking together, which allows towering trees to pull water hundreds of feet up their trunks, completely defying gravity. And incredibly, it's actually less dense as a solid. That means ice floats, which insulates all the aquatic life below it during harsh winters.
All right, Section 2, Energy and Storage Molecules.
So, moving on from our vital solvent, we need to look at our fuel sources. And what we see here brilliantly illustrates a core rule of biology: form follows function. Notice how the alpha-glucose molecules link together to form starch and glycogen. The bonding angles give it this dense, coiled, almost spring-like structure. It's absolutely perfect for compacting and storing energy in a really tiny space inside your cells. But then, compare that to cellulose down at the bottom. Made of beta-glucose, it forms long, straight, unbranched chains. These straight chains cross-link to provide the tough, rigid structural support needed for plant cell walls. It's crazy. We're looking at the exact same basic ingredients, but they yield completely different structural outcomes.
We actually see a very similar principle with lipids, or fats. The crucial point here is just comparing these lipid tails. Saturated fatty acids don't have any double bonds. Their carbon chains are totally saturated with hydrogen, making them straight. Because they're straight, they pack tightly together. Think of butter, which is solid at room temperature. But if you throw in just one double bond, you get an unsaturated fatty acid. That double bond literally creates a kink or a bend in the tail, which completely prevents them from packing tightly. This single molecular kink changes the physical properties entirely, turning it into a liquid oil.
Moving right along to Section 3, the protein building blocks.
Okay, we have the environment, we have the energy, now let's build the dynamic workers of the cell. Every single protein worker in your body is made up of simple monomers called amino acids. Look at this basic structure. They all share the exact same backbone, an amino group on one side and a carboxyl group on the other. But notice that variable R group sticking out? That is the wild card. There are 20 different R groups, and that unique identifier dictates exactly how the amino acid will behave. It decides whether it's going to be acidic, basic, water-loving, or water-repelling. Now here's the question for you. When your cells build proteins, they link these amino acids together in long, straight chains. But a straight chain doesn't actually do anything useful in your body. So how do these simple linear amino acid chains fold into the complex, highly specific 3D shapes required to become, say, the digestive enzymes in your stomach, your immune antibodies, or your moving muscle fibers? Well, here is how it builds step by step.
It starts with the primary structure. That's just the exact, specific sequence of those amino acids. Because of hydrogen bonding between the backbones, this chain immediately begins coiling into an alpha helix or folding into a beta-pleated sheet, and that's our secondary structure. Then those unique wildcard R groups we talked about start interacting. They attract and repel each other, folding the whole thing into a complex, final 3D shape called the tertiary structure. Sometimes, several of these 3D structures even lock together to form a massive quaternary structure, kind of like the hemoglobin that's actively carrying oxygen through your bloodstream right now. The vital takeaway here is that the primary sequence strictly dictates that final, functional 3D shape.
Section 4. The Genetic Instruction Manual.
So we know how the protein workers are built, but who's actually giving the orders in our cellular factory? How does the cell know which sequence of amino acids to string together in the first place? Well, those instructions are held in nucleic acids, specifically DNA and RNA. Think of DNA as the master blueprint. It's double-stranded, incredibly massive, contains the sugar deoxyribose, and uses the base thymine. Because it's so large and so precious, it never leaves the safety of the cell's nucleus. RNA, on the other hand, is the versatile messenger copy. It's single-stranded, much smaller, uses ribose sugar, and swaps out thymine for a base called uracil. Because it's smaller, it can easily slip out of the nucleus to deliver the instructions straight to the factory floor.
Look at how perfectly engineered this DNA master blueprint really is. It consists of two anti-parallel strands, meaning they run in completely opposite directions. The rungs of this twisted ladder are formed by complementary base pairing. Adenine always exclusively pairs with thymine, and cytosine always pairs with guanine. These precise pairings are locked together by those trusty hydrogen bonds we learned about earlier, which creates that iconic, highly stable double helix.
But how do you actually read this ladder? The rules of the genetic code are strict, but they're brilliant. The sequence of bases is read in triplets, called codons. Basically, 3 letters equals 1 amino acid. It is non-overlapping, meaning you read 3 letters, then you read the next 3, without sharing any basis. Interestingly, the code is what we call degenerate. Since there are 64 possible triplet combinations, but only 20 amino acids, most amino acids are coded for by more than one triplet. This acts as a really clever buffer. A small typo might still result in the exact same amino acid being placed. And profoundly, this code is universal. Whether you're a bacterium, an oak tree, or a human being, the triplet UAU codes for the exact same amino acid, tyrosine. No exceptions. And finally, Section 5. The Protein Synthesis Factory.
We've got all the pieces on the board now, so let's watch these molecules actively manufacture proteins in real time. Think of the nucleus as the boss's office, where that sacred DNA manual is kept under lock and key. A specific section of DNA, a gene, is copied into messenger RNA, or mRNA. That mRNA acts as the disposable photocopy, leaving the nucleus through a tiny pore and traveling out into the cytoplasm, straight to the factory floor, which is the ribosome.
Let's zoom in on that copying process for a second. We call it transcription. An enzyme named RNA polymerase literally unzips the DNA double helix right at the start of a gene. It then moves along the template strand, lining up free-floating RNA nucleotides using that complementary base pairing we talked about. If it sees a C on the DNA, it adds a G. If it sees an A, it adds a U. Once the whole gene is transcribed, the mRNA detaches and the DNA safely zips right back up like nothing ever happened.
Finally, we reach the actual assembly line, translation. Out at the ribosome, the mRNA feeds through like a ticker tape. Molecules called transfer RNA or tRNA arrive carrying specific amino acids. Each tRNA has an anticodon, a triplet of bases that perfectly complements the mRNA codon. The ribosome reads the mRNA, the tRNAs match up their anticodons to the sequence, and the ribosome links those adjacent amino acids together with peptide bonds. The ribosome just keeps moving down the line, and out pops a perfectly sequenced polypeptide chain, ready to fold into a 3D protein.
It really is a remarkably beautiful, continuous mechanical process, and it's happening inside your cells right now. But let's pause and bring the energy down just a little bit. Given the millions of bases being constantly copied every single day, what happens when just one single letter in this massive genetic code changes?
Well, we discussed earlier that the primary sequence of amino acids strictly dictates the final 3D shape. So a random spontaneous mutation, just a tiny typo in the DNA, can change the mRNA codon. That typo brings in the wrong tRNA, which drops off the wrong amino acid. And because that new amino acid has a totally different R group, it interacts differently. The protein folds incorrectly. And in biology, you have to remember, if you lose the shape, you lose the function. That one single letter can collapse the entire 3D structure of our protein workers, causing a severe disease or just a complete cellular malfunction.
Yet, without those very mutations, we wouldn't have any variation, adaptation, or evolution. It is the ultimate biological paradox. Considering the immense complexity and microscopic precision required for protein synthesis, how does life balance this razor's edge? How does it balance the absolute necessity for perfect replication to survive today against the need for random mutations to evolve for tomorrow? I'm going to leave you to ponder that incredible thought. Thank you so much for joining me on this explainer, diving into the recipe of life. Keep questioning and keep learning. Catch you next time.